Okay, guys, welcome to day 2 of our online dual credit Anatomy and Physiology 1 class. I hope that you got all the work for yesterday done and submitted to me last night before midnight. And we're going to begin with our last part of chapter 9. This is part 4. And make sure that you have answered the chapter 9 part 3 entrance slip over the stuff that we talked about yesterday before you watch this video. And then again, don't forget that when you finish this video, there will be some questions for you to answer over the stuff in this video. Tomorrow, which will be Wednesday, you will have a quiz over chapter 9. You will and then we will talk about just a few pages in chapter 10, and you'll have some questions to answer over that afterward. In addition, I will go over some things that you will want to make sure that you understand before the exam, and then your exam 3 will post on Thursday, and that exam will cover chapters 7 through 10. And it will be due, all the work that I assign will be due by 11:55 the night that 11:55 at night of the day that I posted it. Sorry, I just got choked a minute ago and I'm having a little bit of trouble. So we're going to go ahead and start them with the last part of chapter 9. So this will be it for chapter 9, and we'll have a quiz over chapter 9 tomorrow, which we'll post on Google Classroom. Today, we are going to be looking at where do muscles get their ATP that they need for muscle contraction. We're also going to be looking at muscle fatigue, and we will also be looking at how the contraction of cardiac muscle and smooth muscle is different from skeletal muscle, which we have spent most of this chapter on. So where do our muscles get the ATP that they need for contraction? Well, there are 4 processes that our muscles can use to produce ATP that they need for contraction. Remember, ATP, that T stands for tri. So ATP is adenosine triphosphate, which basically means that it has 3 phosphate groups. So one process that your muscle can use to get ATP is using the enzyme adenylate kinase, also called myokinase or myosin kinase. Basically, what this enzyme does is it takes 2 ADP molecules, which D stands for di, so this is 2 2-phosphate molecules. So 4 phosphates total, 2 2-phosphate molecules, and it converts it to 1 ATP, which is a 3-phosphate, and then 1 AMP, M meaning monophosphate. So again, myosin kinase or myokinase converts 2 ADP molecules into 1 ATP and 1 AMP. And in this way, you end up with 1 ATP molecule. For every 2 ADPs, you're going to end up with 1 ATP. Another process involves another enzyme, creatine kinase. This enzyme actually takes a phosphate from a molecule called creatine phosphate, and it takes a phosphate molecule from creatine phosphate and adds it to an ADP molecule, remember, which has 2 phosphates. So it takes this phosphate from creatine phosphate, adds it to ADP to form ATP. And in the process, not only do we get an ATP molecule, but we also get a creatine molecule, because when you remove a phosphate from creatine phosphate to turn ADP into ATP, then you also get a creatine molecule as a byproduct. The last 2 are processes that I know you have heard of before. This is going to be anaerobic and aerobic respiration. Again, anaerobic respiration occurs in the absence of oxygen. That's what anaerobic means, without oxygen. And this is mainly going to be used when you're doing brief, powerful contractions. Again, short, intensive exercise. So short term, because when we break down the glucose to produce ATP, we get lactate, which is lactic acid. And so again, the buildup of lactic acid in your muscle cells leads to cramping and muscle fatigue. So you can't do this kind for very long. In addition, you're gonna use this process only as a last resort. So if you're doing a very powerful, intensive exercise, intensive movement, intensive workout, and the aerobic respiration is not producing enough energy for what you need at that moment because you're doing such a powerful, intensive workout, then anaerobic respiration will be used on top of the aerobic respiration. And so again, when you start doing the anaerobic respiration, you're going to produce lactic acid, which is this lactate. It's going to build up in the muscle cell, and it's going to cause cramping and muscle fatigue. And so you can't do it for very long. You're going to be able to get ATP more quickly using this process, but again, it's not as efficient. You don't get as much ATP using anaerobic respiration as you do with aerobic respiration. Which brings us to aerobic respiration. Most of the ATP that our muscles use comes from aerobic respiration. It requires oxygen, again, aerobic, and it breaks down glucose to produce ATP, carbon dioxide, and water. Using aerobic respiration, we're going to produce a lot more ATP. But it is going to be produced more slowly. So this is going to be, again, being produced under normal conditions, normal exercise, when you are not doing this really intensive workout. Normal pace, you are going to steadily produce this ATP. It's going to be produced more slowly but steadily for this normal normal intensity workout. Again, you do get more ATP with this process, so it is more efficient than anaerobic respiration, and it does occur in the mitochondria. You learned this when you were a sophomore in biology, that the mitochondria is the powerhouse of the cell. This is where the aerobic respiration is occurring, using oxygen to break down glucose to produce lots of ATP. This anaerobic respiration does not occur in the mitochondria. It actually occurs in the cytoplasm of the cell. So again, here is a visual to kind of help you see the different ways that muscle cells can produce ATP. Again, we can use the myosin kinase or the myokinase, the adenylate kinase, where we take two 2-phosphate molecules, which are the ADPs, and they are used to produce one 3-phosphate molecule ATP and then one 1-phosphate molecule. So you have 4 phosphates here, you have 4 phosphates here. We have just rearranged them so that we have one 3-phosphate molecule ATP and then one 1-phosphate molecule AMP. The second way we talked about was where creatine kinase, or creatine kinase, I should say, takes the phosphate from creatine phosphate and adds it to an ADP molecule, producing an ATP, and in the process, what we have left after we remove the phosphate from the creatine phosphate, we have just creatine left. Again, the anaerobic pathway, we are breaking down glucose without oxygen during glycolysis, producing pyruvic acid, which is then converted to lactate or lactic acid. Again, the buildup of that lactic acid in muscle cells causes cramping and muscle fatigue. So this is only going to be done during a very brief, intensive workout because you can't do this for very long because the lactic acid will build up, causing cramps and fatigue, and you have to stop. The aerobic pathway is what's going to be used during most normal conditions, normal exercises, where oxygen is used to break glucose down into carbon dioxide, water, and ATP. Again, this produces a lot more ATP than anaerobic respiration. It is a lot more efficient. So again, this happens in the presence of oxygen. That's why it's called aerobic. Okay, moving on and looking at muscle fatigue, when we do anaerobic respiration, we build up lactic acid, and that leads to cramping and muscle fatigue, which when your muscles are fatigued, they are going to be sore. Again, that is due to the damage to the muscle due to the buildup of the lactic acid, which also results in inflammation. And what we end up with is a decreased capacity for our muscles to contract and to perform. There are two types of muscle fatigue. There is physiological fatigue and psychological fatigue. We're going to mainly be looking at physiological fatigue. Psychological fatigue, again, just is caused due to the emotional state of the individual. Can cause contractions, even contractions the person doesn't even realize. Again, they're stressed, they're tense, they have anxiety, maybe depression, and the muscles are tensed without them even realizing it, and so they're contracting their muscles constantly, which can end up in causing fatigue. Tired muscles, sore muscles, fatigue from all the contraction just due to the emotional state of the person. Physiological fatigue is what we're looking at when we are doing these very brief, intensive exercises, workouts, movements. You end up using all the ATP that's available, ATP depletion, so we have to start doing anaerobic respiration on top of the aerobic that we're already doing. This results in the production of reactive oxidative species, which cause oxidative stress and also inflammation. And then again, as I said, due to the oxidative stress, we're going to end up with inflammation, damage to the muscle fibers, and soreness as a result. Okay, so first looking at the ATP depletion resulting in acidosis. Again, when we do anaerobic respiration, it's done to produce more ATP than what we're already producing, especially when oxygen levels are low. As a result, we're going to produce lactate, which is lactic acid, resulting in a lower pH. This lower pH actually affects the ability of the myosin heads to form cross bridges with the actin. Okay, again, remember pH affects enzymes. Well, remember that myosin head is an ATPase. It's an enzyme. So when the pH levels get lower, because of the lactic acid that's being produced, then the myosin heads don't function as well as they could. And so we get weak formation of cross bridges of those myosin heads attaching to the actin. Also, it affects the release of calcium from the sarcoplasmic reticulum. So we're not going to have as many active sites on the actin being exposed so that the myosin heads can bind. And so again, this is going to, in addition to causing the muscle fatigue, it's going to affect the ability to contract of the muscle because of the weak cross-bridge formation and the fewer active sites on actin being exposed due to the effect of less calcium being released from the sarcoplasmic reticulum. The oxidative stress that we mentioned earlier is due to the buildup of excessive reactive oxygen species, which we're going to abbreviate those ROS, reactive oxygen species. These are also known as free radicals. These include things like peroxides. These things are not only produced as a result of anaerobic respiration, but they're also just byproducts of metabolism. And this is why we take antioxidants or eat foods that are high in antioxidants, because antioxidants actually help to neutralize these free radicals, these reactive oxygen species like peroxide that can do damage to our cells, including damage to our muscle fibers. And what these reactive oxygen species do is that they trigger the production of immune system chemicals, specifically a chemical known as IL-6 or interleukin-6. And this immune system chemical is actually involved in inflammation. And it is believed that this chemical is actually the cause of the inflammation and the sore muscles. So again, when we have this oxidative stress, we have this reactive oxygen species being produced, we have our immune system producing this chemical IL-6, which is believed to cause the inflammation and the sore muscles as a result of the muscle fatigue. And then lastly, not only is that IL-6 going to be involved in the inflammation process, but we're also, whenever you exercise, it actually triggers your immune system. And this causes white blood cells known as T cells to migrate heavily into the muscles that you're working out. And this causes increased pain, which alerts us that, hey, we may be doing too much. And we can actually end up, when we do too much anaerobic respiration, we end up with the acidosis, due to the ATP depletion and the doing of and the performing of anaerobic respiration to produce the extra ATP that we're needing. We end up with the oxidative stress from the reactive oxygen species that are being produced and the triggering of the production of the IL-6. And then we end up with the inflammation. It can actually lead to physiological contracture. And this is a state of fatigue where you have a lack of ATP so that you cannot contract or relax. Which, again, we watched a video on this last year where you saw these people running the marathon, and they just had this really weird gait, this weird running that they were trying to do. They kind of looked like, I can't even remember what we said they looked like, where their legs were bent and they were kind of hunkered over and they were lifting their feet really high, and then all of a sudden they just went down, and they tried to get back up and they couldn't, and they tried to get back up and they couldn't. Okay, that was physiological contracture, a kind of fatigue where you have just gone through so much ATP depletion, oxidative stress, and inflammation to the point that you can't contract or relax. You can't really do anything anymore. So what's the benefit of all this? Because obviously this causes our muscles to not be able to work. It causes pain. What is the benefit of this? Well, the benefit of this muscle fatigue is that it prevents your muscles from completely using every single bit of the ATP that they have, and it also prevents very severe damage to the muscle fibers. If it weren't for this muscle fatigue causing the inflammation, the pain that you do have, the cramps and the pain that you do have, then you might use your muscles to the point that they would be severely damaged, which is not something that you want to do, because that would involve a lot of healing time, and I guess could possibly even cause death of the cells. We talked about rigor mortis before. Again, it is very similar to physiological contracture, except, of course, again, both of them are going to result in like very stiff muscles that you can't relax or contract, so you really can't use them at all. The difference is you can still do a little bit of movement with physiological contracture, whereas with rigor mortis, obviously you're dead and you're not going to be doing any movement at all. But basically what you end up with, you have very stiff, rigid muscles, in this case after death. And the reason for this is that after death, calcium leaks out of the SR into the sarcoplasm, attaches to the myosin. It actually attaches to, it's not attaching to the myosin heads. We should say there that it's obviously attaching to the troponin, which then causes the tropomyosin to remove the myosin heads from the actin, so that myosin attaches to actin and cross bridges form. And again, remember, ATP is required for that myosin to release. Well, since there's no ATP being produced, there's no ATP available, the myosin there's no ATP to attach the myosin head, so the myosin cannot release from the actin. And so therefore the muscles get rigid and stay rigid. And then again, the muscles start loosening up and the stiffness goes away several more hours after death. Not because, okay, all of a sudden we have ATP available, but because the tissues are starting to deteriorate. Everything is starting to break down. So the tropomyosin, troponin, actin, myosin, they all just kind of start falling apart from one another. Okay, we also can look at the breathing that we do when we're exercising, especially when we're exercising fairly heavily. And we can look at the breathing that we do at the beginning of our exercise, as well as the breathing that we do after exercise is over. So if you think about it, when you first start exercising, you're not really breathing heavy or breathing hard or fast. But after you've been exercising for a little bit, you start breathing really hard and heavy and fast. And then even after you stop the exercise, you still continue to breathe kind of hard and heavy and fast for a little bit. So why is this? Well, this is what we're looking at now. When we look at oxygen, we call this oxygen deficit, and then post-exercise oxygen consumption. And again, this is referring to the oxygen that we take in when we're breathing, oxygen intake. Basically, those two things are changes in breathing pattern that we experience as we exercise, and that reflect a muscle's need for more oxygen to produce more ATP through aerobic respiration. Oxygen deficit is actually the lag time, or it's the time between when you begin your exercise until you start breathing more heavily. So, as I said, when you first start exercising, your breathing really doesn't change. But after maybe a few minutes of exercising, then that's when your breathing really starts getting hard and heavy. And so oxygen deficit is basically what you're in there, because as soon as you start exercising, you start needing more oxygen to produce more ATP, but your body doesn't realize that yet. So you're just breathing normally. So you end up developing an oxygen debt or an oxygen deficit, which is basically you are needing more oxygen than what you're taking in. You're not taking in, you have insufficient oxygen consumption relative to the activity, relative to what you need to do the physical activity. So that's called the oxygen deficit. Then the excess post-exercise oxygen consumption is the breathing from the time you quit exercising until your breathing returns to normal. And again, this reflects the fact that we have this oxygen deficit here at the beginning. We still are needing more oxygen to produce more ATP because we're kind of behind because we didn't start that hard, heavy breathing when we first started exercise. So even after we quit exercising, there is a time period there where we are still breathing heavily, even though we're not doing that heavy exercising. And that is because we are having to make up or repay this deficit that we incurred at the beginning of exercise when we were doing the hard exercise, but our breathing hadn't caught up to the fact that we were needing more oxygen to produce more ATP. So the lag time between when you stop the heavy exercise and your breathing returns to normal is called the excess post-exercise oxygen consumption. Okay, so, and that is all we're going to be looking at for skeletal muscle. Now we're going to start looking at smooth muscle and the differences between the contraction of smooth muscle compared to skeletal muscle. And then at the end, we'll spend just a little bit of time looking at the differences between cardiac muscle and smooth and skeletal muscle. So again, remember that smooth muscle is non-striated. It has no striations. The fibers are smaller. They have a spindle shape and they have a single nucleus. When we compare smooth muscle to skeletal muscle, we see that there are fewer myofilaments, which means there's fewer actin and myosin filaments overall, and there is more actin than myosin. And we don't have sarcomeres in our smooth muscle. Rather, we have loose bundles. In our smooth muscle, we also do not have T-tubules. We have these things called calveolae. These are indentations in the cell membrane of the smooth muscle, indentations in the sarcolemma that basically are going to do the same function as the T-tubule. They allow the muscle contraction to get down into the muscle cell, and then they also allow calcium to come in from outside the cell, because we're going to see that in skeletal muscle, all the calcium that we need is stored in the SR, but that's not true for smooth muscle. Smooth muscle SR does have some calcium stored, but a lot of the calcium that we need for muscle contraction actually comes from outside the cell. Extracellular calcium comes into the cell to cause the contraction to happen. And so therefore the T-tubules allow, more than anything, they allow, well, not really T-tubules, but the calveolae allow the calcium, the extracellular calcium, to get into the cell. Also, in your smooth muscle, you have these things called dense bodies. In our skeletal muscle, we had sarcomeres, and at both ends of the sarcomere, we had Z-discs or Z-lines, and that's what the actin was attached to. But in smooth muscle, you don't have Z-discs or Z-lines. The actin is actually attached to dense bodies, okay? And then you also have in smooth muscle something called intermediate filaments. These are protein filaments that are not found in smooth muscle, or I'm sorry, they're not found in skeletal muscle. They're only found in smooth muscle, and these do not contract. Okay, so they don't really change position. They don't really do any sliding or anything. They are non-contractile. Also, in our smooth muscle, we see that our smooth muscle contracts a lot slower than our skeletal muscle. The reason for this, remember, as I said earlier, skeletal muscle has all the calcium that it needs for contraction stored in the SR. Once the action potential gets to the SR, this causes the release of calcium. Calcium channels in the SR membrane open, and calcium comes out into the cell, into the sarcoplasm, and attaches to the troponin. Well, in smooth muscle, that you have a little bit of calcium stored in the SR, but a lot of the calcium is outside the cell. And so the reason that smooth muscle is slower to contract is because it takes longer for the calcium to move from outside the cell to inside the cell to cause the contraction to happen. So because the calcium is having to move a longer distance from outside the cell to inside the cell, then this causes the smooth muscle to contract at a slower speed. Also, in skeletal muscle, remember that calcium binds to troponin. Troponin moves tropomyosin away from actin's active sites so that myosin can bind to actin. Well, in smooth muscle, there is no troponin. Calcium binds to calmodulin. Calmodulin serves as kind of an on-off switch to either get the contraction going or to inhibit the contraction. So when calmodulin is not bound by calcium, then the contraction will be inhibited. But when the calcium comes from outside the cell to inside the cell, the calcium binds to calmodulin, and this kind of turns that calmodulin on. What that calmodulin will then do is that it will go activate myosin kinase, or that myokinase that we talked about earlier. And then this myosin kinase will produce the ATP that will then activate the myosin head. So that the myosin kinase is in effect going to activate the myosin head. And once the myosin head is activated, then it will bind to the actin, and cross-bridging will begin, and sliding will happen, and the muscle cell will get shorter. The smooth muscle cell will get shorter. So again, a little bit different method by which the muscle contracts. And I actually have a short video on here. I'm not going to show it again for copyright reasons, but I would definitely take a look at that video. It really is like a 1.5-minute video that really goes through this process, and the guy on there actually kind of draws it out and shows you a little bit more about what's happening. So rather than just reading about it and listening to me talk about it, you can actually see a little bit of visual of it. The only thing is the guy that's talking on the video is a foreigner, so he doesn't call it skeletal muscle. He calls it skeletal muscle. So when he says skeletal muscle, he's talking about skeletal muscle. Again, he just pronounces it a little bit differently than we do. So again, the contraction is slower because calcium has to come from outside the cell to inside. And then once the calcium gets inside, then it's going to bind to calmodulin. Calmodulin will activate myosin kinase. Myosin kinase will then activate the myosin head. The myosin head will then bind to the actin, and the actin will slide past the myosin, and the muscle cell will shorten. So again, we don't have sliding of thin past thick in the sarcomere. We have sliding of thin past thick just in the cell. And we'll see that... we'll look at a picture in just a minute, and we'll see how this is all kind of put together. Also, remember that our skeletal muscles are stimulated to contract by nerve cells, by motor neurons. Okay, smooth muscle can be stimulated to contract not only by nerve cells, by neurotransmitters like acetylcholine coming from nerve cells, but it can also be stimulated to contract by hormones that are being released by glands of our endocrine system. Also, relaxation is a little bit different for our smooth muscle. Remember that in skeletal muscle, or skeletal muscle, as that guy calls it on the video that I'm wanting you to watch, relaxation begins when the ACHAs in the synaptic cleft starts to break down the ACH, the acetylcholine. Well, we don't have ACHAs allowing relaxation in the smooth muscle. We have another enzyme called myosin phosphatase. Myosin phosphatase is the enzyme that's going to result in relaxation in the smooth muscle. If myosin phosphatase is acting while myosin is still attached to actin, then you end up with what's called a latch period, which is a period of sustained tension in the smooth muscle. And then also relaxation will occur due to whenever the calcium starts moving. I don't know if it's pumped out or diffuses out, but anyway, when it goes back outside of the cell, lower intracellular levels of calcium will also result in relaxation. So here we can see the much smaller smooth muscle fibers with their single nucleus and that kind of that tapered end that they have, which is different from skeletal muscle. Again, they're smaller, they're tapered or fusiform or spindle shaped, and they have a single nucleus. Okay, here we can see a little bit more of what I was talking about the differences in structure and function between smooth and skeletal muscle. Here we see the dense bodies. Again, the dense bodies, the purple lines are the actin filaments. So rather than being attached at the end at Z lines, which is true for sarcomeres, we don't have any sarcomeres here. What we have is we have the actin filaments attached at each end to dense bodies. And then we can see the green filaments are the thick filaments. And so when the calcium comes in and activates the myosin kinase, which then activates the myosin head, then the myosin heads will attach to the actin and we will get the sliding of the actin past myosin. When that happens, it pulls those dense bodies similar to the way it pulls the Z lines closer together, shortening the sarcomere. We see the dense bodies being pulled closer together and shortening this this smooth muscle cell. Also, you can see here these blue lines are those intermediate filaments. As you can see here in both pictures, they are not getting shorter. The intermediate filaments are not getting shorter. They are non-contractile. So they don't get shorter. Basically, they just kind of are moved as those thin filaments slide past the thick and those dense bodies are brought closer together. So we kind of have what we see kind of a squeezing here, but the mechanism by which the squeezing happens is different in the smooth muscle as compared to the skeletal muscle. Again, so the little dots here are the dense bodies that the purple thin filaments or actin filaments are attached to at either end. The green are the thick myosin filaments. When the calcium binds to calmodulin activates myosin kinase activates the myosin heads, and so then the myosin heads will attach to the actin and slide the thin past the thick, resulting in these dense bodies moving closer together as the thin slide past the thick. And effectively, when that all happens all over the cell, we get the squeezing or the shortening of that smooth muscle fiber. Also, we can see here the blue intermediate filaments not shortening, not contracting, but just being moved as those dense bodies are pulled in closer together. This is the video I was talking about. Again, I'm not going to show it to you on my video because I don't want to get flagged for a copyright infringement. But if you will go to the PowerPoint on Google Classroom and you will find this slide and click on this video. It's about a 1.5-minute long video, and it will kind of give you a little bit of a visual. It will go over what I've talked about, but it will give you a little bit more of a visual because I know most of you are probably visual learners. Okay, just going through those steps again. These first two are just talking about the stimulation of the smooth muscle cell. Again, normally when we talk about skeletal muscle, we have depolarization of the plasma membrane, the sarcolemma, as the action potential is transmitted from the neuron, which is the presynaptic membrane, to the postsynaptic membrane by that acetylcholine, that neurotransmitter. So if you have a neurotransmitter that is, acetylcholine, and you also, I think there's another neurotransmitter that can stimulate smooth muscle cells. When you have a neurotransmitter stimulating, sending that action potential, causing depolarization of the plasma membrane, it's pretty much like a skeletal muscle as far as how that happens. But also remember, not only is smooth muscle stimulated by neurotransmitters, by, causing action potentials, but hormones can also stimulate and cause action potentials in smooth muscle. So that's what this first one was talking about. When a hormone combines with a receptor, this activates a G protein mechanism, which we're not going to talk about that G protein mechanism right now, but it does involve an alpha subunit. So if a hormone attaches to the plasma membrane, the sarcolemma of the smooth muscle cell, then this activates a G protein mechanism, which contains an alpha subunit. This alpha subunit will open up calcium channels in the sarcolemma, allowing calcium to move from outside the cell to inside the cell to bind to that calmodulin. Whereas if we have a neurotransmitter binding to a channel protein on the smooth muscle cell membrane, the sarcolemma, then that's going to open up sodium channels and allow sodium in, which is going to deliver a an action potential. It's going to cause depolarization of the plasma membrane. And in either case, whether we have an alpha subunit, whether we have a hormone binding, activating a G protein mechanism, an alpha subunit of that G protein mechanism, opening calcium channels, or whether we have a neurotransmitter binding, opening up sodium channels. In the end, we're going to, whether we have the G protein mechanism or the depolarization, in either case, we're going to open up calcium channels. So both of those, whether it's a hormone or a neurotransmitter delivering an action potential, in either case, we're going to end up opening up calcium ion channels. Calcium ions will diffuse from outside the cell to inside the cell, and then those calcium, and here's the calcium ions. So here we have the calcium ions coming through a calcium channel that has been opened by this G protein mechanism. Here we have calcium ions coming into the cell from outside the cell. Actually, this is still the G protein mechanism, but in either case, it could also be due to depolarization, the calcium ion channels open. In either case, calcium ions come into the cell, they bind to calmodulin. Calmodulin activates myosin kinase right here. This is the myosin kinase. So calcium, the green dot, binds to the calmodulin, the purple square. It activates myosin kinase, which is the red kind of square, and then the myosin kinase will activate the myosin head. The myosin head will attach to the actin filament, sliding of the actin past the myosin, and shortening of the muscle cell. And that's what all this is talking about here. So again, I've highlighted the major parts of it. Okay, we have two types of smooth muscle. We have visceral smooth muscle and multi-unit smooth muscle, and they are different from one another. Visceral smooth muscle is the most common type of smooth muscle that you have. It occurs in sheets. It's going to be present in places like your digestive tract, your reproductive tract, and your urinary tract. There are gap junctions between the cells. Again, remember, gap junctions allow communication, so they allow the action potential to pass from one cell to another in this visceral smooth muscle. And this allows the sheets of muscle to act as a unit, to all contract together. Often, this visceral smooth muscle is autorhythmic. For example, the visceral smooth muscle in your digestive tract is autorhythmic. So, it really doesn't have to be stimulated in order to contract. These gap junctions can just pass the contraction from one cell to the other, causing the whole sheet to function as a unit and to contract rhythmically. But it can also be stimulated. Some types of visceral smooth muscle, such as that in the urinary bladder, only contracts when stimulated. So again, this autorhythmic means automatically contract without stimulation, at least without conscious stimulation. And then this is, or I should say without stimulation, because only skeletal or voluntary muscle is consciously stimulated. But anyway, it can contract without stimulation. That's what autorhythmic means. It's contracting without stimulation by the nervous system. But then it can also be stimulated by the nervous system, such as that in the urinary bladder, and contract in the same way. And then we have multi-unit smooth muscle, which can occur in sheets, bundles, or a single cell. So sheets, it occurs in sheets in your blood vessels. It occurs in bundles, such as your erector pili muscles in your skin are bundles of smooth muscle. And then it can also occur as single cells that act independently, such as smooth muscle in the capsule that surrounds your spleen. Again, this multi-unit smooth muscle generally only contracts when stimulated, so it is not autorhythmic. It only contracts when stimulated either by nervous mechanisms, that's going to be your neurotransmitters like acetylcholine, delivering action potentials, or by hormones. Okay, some differences between the electrical properties of smooth muscle and skeletal muscle. Remember, skeletal muscle, I'm going to go ahead and fix that there. In skeletal muscle, the resting membrane potential is around negative 90 millivolts, but in smooth muscle, it is around negative 55 to negative 60 millivolts. Also, remember, skeletal muscle contracts very quickly, whereas with smooth muscle, we have slow waves of depolarization and contraction. Again, with skeletal muscle, the depolarization is caused when the acetylcholine attaches to the sodium ion channel and causes it to open. allowing sodium to come in. Well, with smooth muscle, again, the depolarization is caused by the diffusion of both sodium and calcium into the cell. So again, not only do we have the neurotransmitter binding to sodium ion channels, allowing sodium in, but then that also opens up calcium ion channels that allows calcium in. Remember, we need that calcium inside the cell so that it can bind to calmodulin. It can activate calmodulin, which will activate myosin kinase, which will then activate the myosin head of the thick filaments. So you have the diffusion of both sodium and calcium into the cell when depolarization occurs. That's what causes depolarization. Also, similar to your your cardiac muscle, smooth muscle can have pacemaker cells. So again, it is autorhythmic. It has its own cells that develop action potentials and cause the contraction rather than being stimulated by neurotransmitter, by nervous mechanisms or hormone mechanisms. So again, it is autorhythmic because of those pacemaker cells. And then again, as we said earlier, the contraction can be stimulated not just by neurotransmitters, by the nervous system, but also by hormones. Again, a lot of this we've already talked about. Differences in the functional properties of smooth muscle. These are properties not seen in skeletal muscle. Again, smooth muscle is autorhythmic. It can be stimulated to contract by the pacemaker cells in the smooth muscle. So no nervous stimulation is necessary in order to cause the contraction of the smooth muscle, whereas skeletal muscle only contracts as a result of stimulation by the nervous system. Another thing that is different from skeletal muscle, it will contract to sudden stretch, but it will not contract. It will not be stimulated to contract by a slow increase in stretch. So for example, when your stomach feels, when your bladder feels, okay, when your bladder feels as it slowly and gradually feels, it slowly stretches, okay, you can still hold your bladder. You don't start getting that contraction, that autorhythmic contraction, and oh, you've got to go to the bathroom, okay? A slow stretch does not stimulate the contraction of the muscle, but a sudden stretch can stimulate the contraction of the smooth muscle. Also, you have a relatively constant tension in your smooth muscle known as smooth muscle tone, which is different from what we call regular muscle tone in skeletal muscle. Again, in skeletal muscle, some of your motor units are always contracting, and that helps to keep your bones and muscles and tendons and things like that in the correct position to avoid damage to those. In smooth muscle, we have what we call smooth muscle tone, which is a relative constant tension. So it's not some motor units always contracting even when the muscle is at rest, but it's just a relatively constant tension that's always there. Okay? So again, what can stimulate our smooth muscle? Again, as we said, it can be stimulated by not only our nervous system, but also by hormones. So we have actually two different branches of our nervous system that can stimulate our smooth muscle: our autonomic nervous system and our somatic nervous system. When our autonomic nervous system stimulates our smooth muscle, that is involuntary. Our somatic nervous system only stimulates our skeletal muscle, okay? Somatic nervous system stimulates our skeletal muscle, and that is our voluntary muscles. It is our autonomic nervous system that stimulates our smooth muscle, and that is involuntary. So again, remember, smooth muscle is involuntary, and the branch of the nervous system that causes the stimulation of the involuntary responses, involuntary muscle, which would be smooth muscle, would be the autonomic nervous system. It is the somatic branch of our nervous system that stimulates our voluntary muscle, which is our skeletal muscle. Again, remember that the neurotransmitter that stimulates our skeletal muscle, the neurotransmitter that is released by those somatic neurons, is acetylcholine. But with your autonomic neurons, you actually have two different neurotransmitters that can be released. Acetylcholine and norepinephrine. Both of those are neurotransmitters that are released by neurons of the autonomic nervous system. And we'll talk a lot more about the autonomic nervous system and those neurotransmitters. So if you go ahead and kind of grasp that concept right now, that will make some of the things that we're going to talk about later on when we talk about the nervous system a little bit easier to comprehend. It'll be one less thing you'll have to remember because you'll already remember it from here. So again, when our somatic nervous system stimulates our skeletal muscles, we are releasing only acetylcholine. Acetylcholine is the only the only neurotransmitter that stimulates skeletal muscle. But our autonomic neurons, our neurons of our autonomic nervous system, the involuntary a part of our nervous system, can stimulate our smooth muscle but with two different neurotransmitters, acetylcholine and norepinephrine. Again, hormones that are important in stimulating smooth muscle to contract are oxytocin and epinephrine. You may have heard of oxytocin before. Oxytocin is the hormone that is released that causes contraction of the smooth muscle in a woman's uterus when she is in labor and having a baby. So oxytocin is one of the hormones. Now, it's not the only hormone, obviously. There's going to be others, but oxytocin is one important one. And then the other one is epinephrine. Epinephrine is a hormone that can also act as a neurotransmitter, but it is a hormone as well that can stimulate the contraction of smooth muscle. There are various receptors on the sarcolemma, the plasma membrane of smooth muscle cells, and which neurotransmitter or hormone is binding. We have receptors for epinephrine, oxytocin, acetylcholine, and norepinephrine. We have receptors for all of those in the sarcolemma of our smooth muscle cells. So what response the smooth muscle does depends upon which one of those neurotransmitters or hormones binds. So for example, when epinephrine binds, that is actually inhibitory for our smooth muscle. Epinephrine is adrenaline. Okay, it is inhibitory for our smooth muscle. It actually inhibits the contraction of smooth muscle and slows our digestion down during a fight or flight response. Whereas norepinephrine is stimulatory. It activates our smooth muscle to contract. So during our rest and digest response, when we're calm and relaxed, our norepinephrine or noradrenaline is stimulating our smooth muscle to contract so that we can digest our food and we get hungry and we have to go to the bathroom. So again, which neurotransmitter or hormone binds to the receptors on the cell membrane of the smooth muscle determines what response we're going to see, whether we're going to see inhibition of the contraction of the smooth muscle or activation of the contraction of the smooth muscle. Okay, very last thing, looking at a little bit of difference between cardiac smooth muscle and skeletal muscle. Again, remember that cardiac muscle is found only in the heart. It is striated. It usually has only one nucleus, and it is the only one of the three types of muscle cells that are branching so that there's lots of connections between cells, and these connections again or junctions are called intercalated discs. Remember, these are gap junctions similar to the gap junctions that we have in smooth muscle tissue, in the sheets of smooth muscle tissue or the bundles of smooth muscle tissue. These gap junctions are communicating junctions, so they allow ions to move across so that the contraction can be conducted smoothly across the entire sheet of smooth muscle, or in this case across the entire heart muscle. Again, like smooth muscle, cardiac muscle is also autorhythmic. Now remember, not all smooth muscle is autorhythmic. Visceral smooth muscle can be autorhythmic or can be stimulated. But remember, your multi-unit smooth muscle is actually not autorhythmic. It only is activated to contract by stimulation. So our cardiac muscle is autorhythmic, similar to some of our smooth muscles, such as that visceral smooth muscle that's found in the digestive tract. Also, cardiac muscle has action potentials that are longer in duration than that of even smooth muscle, and definitely longer than that of skeletal muscle, and so you have a longer refractory period. And remember, the refractory period is just a period during which the cell membrane cannot respond to sodium ions again. So in cardiac muscle, you have a longer refractive period, a longer period during which the cell membranes of the muscle cells, the cardiac muscle cells, cannot respond to sodium. Again, this is going to allow complete relaxation of the heart muscle before sodium ions can cause it to contract again. And again, that's very important for the movement of the blood into and out of the heart. Like smooth muscle, cardiac muscle is depolarized when both sodium and calcium are moved into the cell. Remember that skeletal muscle depolarizes when sodium ions move in. Smooth and cardiac both are depolarized when both sodium and calcium ions move into the muscle cell. And then calcium regulates the contraction similar to how it is regulated in skeletal muscle. Now, that is different from smooth muscle. Remember, calcium comes in and binds to calmodulin in smooth muscle, but in skeletal muscle, calcium comes in and binds to troponin. So we're going to have a similar regulation of contraction by calcium binding to troponin in cardiac muscle, just like you do in skeletal muscle. So in terms of calcium regulating contraction, it's going to be similar to skeletal muscle. But in terms of depolarization, it's going to be similar to smooth muscle in that both calcium and sodium come in to the cell from outside. Here we have a picture of cardiac muscle. You can see the striations. You can see the intercalated discs, which are those gap junctions that allow ions to move between cells so that contraction can be conducted smoothly across the entire muscle, similar to the visceral muscle in our digestive tract. And then you can also see the branching. Remember that cardiac muscle cells are the only ones that actually branch. So I hope that that you understand this material. Please, again, email me if you have any questions or concerns. Go back and watch the video again. Go back and just watch certain parts of the video again. And don't forget, before you're done with your work for today, you're going to answer the questions on the Chapter 9, Part 4 exit slip and submit those to me. Remember that everything needs to be done for today by 11:55 this evening. And we'll talk to you later.